Babylon? Yes. So I think which has been postponed for quite some time, that what we have tried to do is that the four of us who was part of the original application try to give a short presentation about who we are, our backgrounds, and where we work, and what our plans are within this project, and maybe also if we have done anything related to it. But let me start. So the logo we have seen before, drawn by Eve, I assume, and it says that my name is Christian. I come from the Bioinformatics Research Center, and today is March 1st, and it has been other dates. It's a different version of this presentation, and it all relates to this grant from the Novo Nordisk Foundation that we received last year. And there, this slide we also have seen before. The central part of the slide was on Epic slides about a year ago, and then I guess I concatenated two slides into one, but eventually reminding myself about who we are, and we have just done so physically, and everybody is here except Ariane, who is from Belgium, right? But today traveling around. Victoria, Eve, myself, and then the pictures that was available of people when Eve did his slide a few months ago, or maybe that slide was from last year when you did it the first time. But here it is, and in blue with at least some headlines inspired by what was in the application, saying what we would like to do. And I'm supposed to talk about Johan and myself. And just a few things about the Bioinformatics Research Center. Maybe you know about it, maybe you don't. It has been around our faculty for quite some time. We have moved a few times. Last year, around this time, we moved to where we are now. We all know, maybe you don't, how the AU campus here in this part around us are physically. We are right here, this corner of the campus, like upper left part, of course, depending on where you stand. But iNano is here, and the Bioinformatics Research Center is over here, actually more or less where the arrow points. So this is a large building complex, and the first building complex in this huge, mainly for you, red part, which basically just looks like a red version of what is the yellow university over here. It used to be a hospital, we all know that. And these two northernmost buildings has been was ready for people and use last year around this time, and molecular biology has moved in, and BIRC as a smaller entity and other entities occupies the building. So that's where we are now, and probably also where we will remain for quite some time before we were down here. Anyway, so now you know at least how part of the campus look like from above. And this is, except for the building complex, we are in the two uppermost buildings. It's mainly a construction site where you cannot enter unless you do so in the dark. And the center has been around our faculty for quite some time in various forms. We were established in 2001, and we are still around. And we are currently around 35 researchers, a few 6 with permanent positions, postdocs, PhD students, researchers in a broad sense, associated to the center. Some are there all week, all time. Others are affiliated to it and is there 1-2 days a week and have their other workplace at for example, the university hospital or elsewhere at the Faculty of Health. Backgrounds are quite mixed, reflecting the backgrounds of bioinformatics. We have people who have their education, PhD degree in more biological areas, and some who have in a more computer science or mathematical area. And there's also some bioinformaticians around. But I guess assume like here, you have also various backgrounds from the broader area of biological sciences and life science, and we then also have some with the computational and mathematical sciences. Yeah. And then all years actually since we was established, we have been running or responsible for a master's program in bioinformatics, which currently have about 40 students enrolled, about 20 students in each of the 2 years of a master program. And a large fraction, about half, are international. And again, half of the half are maybe from EU and the other half from non-EU countries. Unfortunately, the program is part of this educational programs where the intake is restricted because the candidates are unemployed. That does not go specifically for bioinformatics, but bioinformatics, probably due to the prefix bio, is part of the administrative group nationally that is the group of biological educations. And many of those, and unfortunately the largest of those, cannot really find employment for their candidates. So we are not allowed to enroll more than 15, 20 students each year, but there are many more who apply. So that's a little bit pity, especially taking the current financial situations into account.
And also because IT, they prioritize that, and actually you are doing.
But that's a story for another time. We were part of that at one point, but then somebody thought we shouldn't. Good. Then a majority of the people at BIRC traditionally and is still working in a more population genomic area of bioinformatics. So many of the biologists who have been around since the center was established had a background in genetics, and that still is a majority of the activity. Myself, I had a background in computer science, and genetics also fit nicely in there because it all strings, so a lot of string algorithms were applicable to that. But there has also, during the time, have been some activities in structure bioinformatics, also RNA structure bioinformatics of various kinds. But Johan and I are those at BIRC that do COFOLD stuff. So we are the COFOLD part of it. And Johan, as he said, he's a PhD student, financed by the COFOLD project, been around since September. And your background when you applied for it was you had enrolled in the master program in bioinformatics, and you was that for about a year, and then you can switch to a PhD program in the Danish system. And before you were master students, you was a bachelor student in molecular medicine, and that study ended with a project supervised by Ebbe. So there was some connections established, and Ebbe and I had also knew each other before for a long time.
I was actually once for a short time hired at the Bioinformatics Research Center.
Anyway, myself, I've been around for quite some time. I have a background in computer science. I have a PhD in computer science in what would then be classified as theoretical computer science, focus on algorithms and data structures, but algorithms and data structures in a more practical setting, because it was mainly targeted toward bioinformatics or computational biology inspired problem, which meant that it to a large part was algorithms dealing with strings, finding patterns, comparing them, and whatnot. After the PhD, had for many years an associate professor at the Department of Computer Science, and later, not that many years ago, when it became possible, those that were affiliated to BIRC but had permanent positions elsewhere at the faculty were kind of, their positions was transferred to the Bioinformatics Research Center. But in everyday life, it's still such that I have connections to the computer science department, but more and more to the Bioinformatics Research Center only. And I have been responsible for running BIRC and our master program for also many, many years. And now I said that Johan and I were the ones who did COFOLD stuff at BIRC. We are, and we're also doing it next semester. But for this semester, we also have a couple of master students who do their projects on topics related to our part of this project. So we have Andreas, who is doing his master thesis in bioinformatics and shares an office with Johan, and doing some work on some of the stuff I will mention later, basically on trying to understand the existing tools, the existing data sets that they claim to have benchmarked on, and basically trying to get a feeling of, do things actually work as claimed? And then there's another guy, Jan Arten, or however you pronounce it. I have only tried to pronounce his first name, and he responds. who's a master student in computer science and more interested in say algorithms and data structures. So we basically do something that I would have done many years ago. You're interested in algorithmic stuff. It's motivating if you can say that the problem you look up is not some abstract graph problem, but you can have an introduction saying that the solution to this also relates to this area. And you also have an interest in implementing the algorithms and examining practically. If you do algorithms in a computer science setting, you might well just be interested in designing the algorithm and investigating it in a theoretical way. But there's, of course, often a difference between theoretical efficiency and how they can be realized on various kinds of computational hardware. And he's interesting. And he's also working on some of the topics that you are interested in and we look at. Namely, how can you compare RNA secondary structures fast and reasonably relevant, such that you can say something about, for example, what is in a database? Is it representative in one way or the other? That is probably what he will end up doing. Right now, he's also looking at if he can re-implement or improve time-wise, efficiency-wise, some of the existing prediction that's been around. But it seems that the comparison of structures and trying to quantify distances in an efficient manner is what he seems to like. Yeah, and then a little bit about my background. I have actually studied, so my interest is design and implementation of both what we call worst case efficient algorithms, or algorithms that are efficient theoretically, and also their efficiency in practice. So how do they behave if you actually implement them? There are many examples of algorithms that are efficient in theory, worst case in some model of a computer, which if realized on contemporary hardware, they are something that doesn't really work, that you need to take into account in bioinformatics. And during the years, that has been sequence analysis, algorithms for comparing or aligning sequences. You all know pairwise alignment, multiple alignment, try to essentially speed it up or make the models, the comparison model, maybe more refined or fine-tuned toward a particular application. Can you still then do it efficiently? Also algorithms for searching large amounts of sequence data to look for specific patterns. It could be patterns that are repeated or have other kind of structures to see if you can do that fast. And also interest in contributions in phylogenetics, improving the speed of construction algorithms, for example, for the very classical construction method, neighbor joining. Can you do it more efficient such that you can construct trees or cluster sets of hundreds of thousands of objects rather than maybe just thousands? And also various methods for trying to fast quantify the distance between two tree structures. It's easy to see if they are completely alike, but if they are not, there exist many measures for how to describe how far away or how similar are they. Some of them you can compute fast, and some of them you cannot, and studied a lot of methods based on basically quantify distances between trees by decomposing them into a set of smaller trees and then compare them. And this is something that maybe also could be used to compare RNA secondary structures, because many methods that compares RNA secondary structures kind of use these secondary structures as a tree topology, so a branching structure, and then compares that in some way where you of course take into account that what you measure should also be relevant if you view the trees as RNA. But sometimes these measures are very slow, and maybe some of these quartet or triplet based methods that they are called could be actually be used. The distances they give between trees could maybe also be a reasonable distance measure if the trees you compared were, let's just call them RNA secondary structure trees. It's essentially, if you move one of these classic RNA secondary structure pictures, it looks like what in computer science and mathematics would be a tree, right? There are some nodes where edges, branches off, and as long as there are no pseudonodes, that kind of makes it not a tree, because things are, then it is, if you take an RNA secondary structure up and it dangles here, it would more or less look like a tree, where the branches would be helices and the various kinds of loops would be things where helices branches off from each other. So that could maybe be used. Also made some algorithmic contributions and still interested in structure prediction of various kinds. Many years ago, the CS field was very much interested in some such very simple cartoon-like lattice models of protein structure, where you said a folding of a protein was a embedding in a grid, for example, like a square grid on a piece of paper. If you could embed it in a self-avoiding way, that was a fold. And that, of course, makes for much simpler models and real-life folding, where it can be, there's a lot of, but people were very interested. How fast can one predict structures in such models? Is it actually hard to predict it in that model? And it turned out to be quite hard, and one could make some theoretical contributions, and many did. But of course, since even ridiculously simple models was hard to formulate, say, a free energy-based prediction for, then maybe the more complex models probably was equally hard, at least. Yeah, and sometimes, of course, you can be cheated by such, because sometimes actually making things more general makes it easier. But also looked at RNA secondary structure prediction, and also, like, again, more like a negative result, proving that at least in some of these theoretical models for RNA secondary structure prediction, it was hard to predict secondary structure, kind of showing that if they included pseudoknots, an arbitrary number of pseudoknots, because anything is easy in a computational fashion if you just have a limited amount. Because if you have a limited amount of stuff, then you can enumerate them all, and then you can at least solve it by looking at them. But had some various proofs that that you shouldn't hope to make a general algorithm that could predict secondary structures with pseudoknots using a reasonably acceptable score function. Of course, prediction is super easy if the score function is for you to decide. Then you could just say, well, any structure scores the same, so finding the best one is easy, just pick one, right? But of course, it's that would make for a ridiculously irrelevant solution. So of course, it's a combination of how the problem is turned into a mathematical question. But we had some. Then I'm also interested in machine learning, and inspired by the other people at BIRC, we were especially interested in say, probabilistic models that you would call machine learning models, like hidden marker models or stochastic context-free grammars, and that can be used for various problems. Hidden marker models for many of these sequence annotation problems, and also stochastic context-free grammars are also related to secondary structure prediction. Basically, what I've done there is, again, with the background algorithms, trying to see if you can, both in theory and practice, speed up the existing algorithms that are used, for example, training models or applying and finding the best possible structure or the best possible annotation. Yes. And what we are trying to do in Johan and I in relation to this project is, in the application it was described like this, right? But in a more cartoon-like thing, how I think what I guides me when we find things to talk about and work on, it's basically trying to find folding algorithms or construct folding algorithms that takes the knowledge of this code transcriptional folding process into account. You can of course say that that is also taken into account by existing RNA secondary structure prediction methods like RNA-fold or M-fold or even old-fashioned Nusinov. Basically, it must be taken into account by any method that produces folds which some people say are good. Because if code transcriptional folding plays a role, then implicitly in the design of these algorithms, and for example, if they were methods based on minimizing a free energy, thermodynamic stuff, then of course the optimization problem must have been formulated in such a way that it also captures the effect of code transcriptional folding, unless of course the examples on which it has been evaluated and where it has been used by various groups and where they're happy of using it are examples or applications where, of course, code transcriptional folding must play a role, because that is how RNA, but maybe not a crucial role for the structure that has been generated, right?
I think the original like work was only thermodynamic and only was based on like folding by heat annealing in a test tube. So where they actually didn't look at biological structures. So that was also why the early paradigm somehow was only fitting like with heat annealing, that somehow the chemist kind of measured all the stacking energies, and then that could kind of be used to predict a thermodynamic most stable model, and then that was then also what you got when you heat annealed the sequence, and there was some correspondence. But then when you went and looked at nature, then actually often that did not match. So.
But it is true for many of the machine learning models that we have. That they are usually like trained on data that has taken co-transcriptional folding into account.
Exactly. Yeah. So
in some way, if they are any good, they will have to take it into account.
So then they probably also spot the effect of that, but of course they don't explicitly model it, and therefore you maybe cannot rip the methods apart and learn something in the other direction about the process. But indirectly, they take it into account if the examples you consider when you sit around and design an algorithm. This is important. But this is what we basically have been looking at, or what we still look at. Some algorithm, some black box that can take a sequence and predict a secondary structure that take co-transcriptional folding into account in a suitable fashion. And that has involves different lines of work. So of course we have started out, and I'm still looking at, and Johannes also, at the existing approaches that builds upon very classic minimized free energy based approaches that claims to take co-transcriptional folding into account. So there's this now call it the old 10-year-old paper called COFOLD that is to extend the existing RNA fold method by the Vienna group by some rules, or you can say additional terms to the function they try to minimize, that and somehow takes co-transcriptional folding into account. It's validated on some examples. And the general idea there is basically that co-transcriptional folding should make it more favorable to make base pair interactions that are not spanning a long part of the sequence. They're probably more likely than those that are shorter, right? Because if a structure is created along the way, then if the last base pairs that is generated has to pair with the first one, then it has to interrupt or maybe disrupt a large already generated structure. So this long range interaction, at least string-wise long range interaction, is probably not that likely to occur, unless of course the very first sequence is unpaired in the structure that has already been generated. But if a base pair is already paired, then maybe the new one or the new ones generated try not to disrupt that. And you can kind of see how that could be maybe formulated mathematically. If the difference between base pair J and base pair I is huge, then that is somehow penalized in the score or the penalty, the inclusion of a particular base pair takes. So it's stuff like that. And I think it's a nice paper, and it's also a simple thing to do. But and this student from the CS department is at least one possibility is that he will try to summarize or give a review of such methods and then implement it and see if he can actually see the same effects computational wise and how the structures fit. And there are several of such papers. So the Vienna group, inspired by this, but maybe as an answer to this, said that their folding approach, the RNA fold, could be generalized or include a lot of additions, like this condition that base pairs cannot be too far apart, or I and J cannot be too far apart. So, which you could call a mathematical constraint. So they have a paper where they show that their methodology or the structure of their algorithm can be extended with all kinds of constraints as long as they obey certain things. So they made a paper about the RNA fold where they kind of described the various constraints that their method could be extended with that wouldn't affect the running time of the algorithm in practice or in worst case. Basically, in a polite way, saying that what you did with COFOLD is just a special case, what we can do with a lot of different things. Right? So that's a typical response in an algorithmic community. Some people do some very practical extension of it and explains why it's good, and then the other maybe feels a little bit offended and said, okay, we can handle your case and a lot of other cases without making the complexity of our algorithm. Because then they have kind of closed the gap for all possible follow-up things.
Yeah, but it's important to say that that doesn't mean that the practical usage can't like improve from actually adding some constraint. It's just usually algorithmic folks aren't that interested because it doesn't really affect the algorithm. It's the same algorithm just with new constraints.
Exactly. Yeah. And I still like this line of work and exploring how much can you do within a certain time frame in theory and practice. And a lot of the things might not have any practical implications at all, but it's always nice to explore the boundary between how much can you do in a certain amount of time or not. Then we've also been looking at, and still quite interested in machine learning based approach for RNA structure prediction, including those that addresses co-transcriptional folding. And there's a fine line between what you would call a classical method and machine learning method, because essentially machine learning methods tries to predict the parameters that are in a formula from existing solution, existing data. So as you ever said, you could make experiments that adjusted a lot of the energy parameters in these thermodynamic approaches. How much should you gain or be penalized for introducing a certain base pair, and or a certain loop, and how much did a certain helix contribute to the free energy or not? You could base that on physical experiments. So you basically learn the parameters of the function, like the 4 instead of 4 times x squared plus 5 times x plus 6. Why should it be 4, 5, 6, not 2, 42 and 13? Well, you could learn them by real world experiments. Machine learning based approaches try to learn these things from examples, at least machine learning based approaches that are used here in this area, which is typically what is called supervised machine learning based approaches, where you from examples of existing, say, sequence, real structure or true structure. If you have a lot of those, and a lot, you could learn the structures of the parameters of a certain kind of function. And many people here in RSA structure prediction uses what's called neural networks, and you have probably heard about them. Many are based on a specific version of neural networks called CNN, and they have been applied with enormous success for image prediction. You all see this. An image, this is, if it was, say, it was a cat, then is it a cat? Yes, no. Then for a lot of examples of pictures where cat, yes, no, you will kind of learn what this function here should look for in order to say yes or no. And that has been reused, and I think in a very simple and elegant way. I don't know how applicable it is in practice for RNA secondary structure prediction, where you basically say, okay, you have a sequence that could be turned into a representation graphically, a picture of all possible secondary structures. Essentially, you have this dot plot of a secondary structure that you've probably seen before, and then you place a dot with where there could be a base pair, and where there are not a dot, it means that the corresponding two bases cannot pair according to the rules. Otherwise, you could place it in a color that represents what kind of base pair it is. So this is not a secondary structure. It's basically a picture of all possible secondary structures. Then in your examples, you have the true secondary structure, which of course is a picture where you could also represent graphically just by, again, this dot plot thing, where you have a dot where for the base pairs where there was. And then if you had enough of this, and you really need a lot, you could say this picture becomes this picture. This picture apparently becomes this picture. So I have a lot of examples, and then these concurrent neural networks are very good at figuring out that function, such that you could basically, if you showed enough examples, you would come with an unknown sequence, as a sequence of unknown structure, show it the picture, and then it would say, the corresponding picture, the corresponding structure, true structure to this is something. So, and that's like a very classical thing, and there's a lot of things that is difficult here. For example, in practice, in their study here, and in many related structures, they don't succeed in making a function that maps it to a real structure. It kind of, you could also say that it cleans up this picture to something else by removing those potential base pairs that actually do not seem to appear. And then they typically end up with something that is almost a structure, meaning that there could be dots, like 2 dots in the same row, that would say that a single base participated in 2 base pairs. That's of course not true, but they end up with something that is close to, and then all these papers, and that's probably where, not the cheating, but where what maybe makes them not that applicable. They have a cleaning up method that is basically try to remove as few, for example, dots as possible that turns this prediction into something that is actually valid. Typically just saying that no base can participate in more than 1 base pair. And of course, a lot of the precision lies in this post-processing, which for many of these approaches are not super precise, because I guess these are made by people who have an interest in machine learning. But I think there's many of such examples. And
how many is a lot?
That's
how many
different.
sequences do you need, do you think?
I don't know.
1 million or 1 billion or
The more the better.
like
but
10 to the 9?
Okay,
to the
so
19, or...
yeah, so yeah, I'm actually looking into that.
Yeah.
And the thing is, like, for now, I think if we had those like 100,000 structures that are in databases around the web, in a non-biased way, I think we would have much better results, at least from these methods, because they would be able to learn something. But we have a lot of structures. Like the main problem that I'm looking into right now is that the sequence space is much bigger than the structure space. So a lot of different sequences maps to the same structure. And that means basically that we have a lot of families in the databases, which are similar in structure, but not necessarily in sequence, because there's a lot of evolutionary distance between them, but they have kept the structure because that is important evolutionary. Yeah, that's why I'm looking into ways of measuring distance between structures, because that is something that we see is not really big in these models. And the thing is, like, these models often get biased towards predicting the mean of a certain family. So it sees a sequence, goes, okay, so that depends, that's in this family, I'll just predict the mean of that family. And that pretty much, that gives a pretty good score compared to other things that we can do in machine learning and like the other algorithmic approaches. But it isn't really, it's just basically just classifying a sequence into a family, which we can already do, like just by looking at the sequence.
So a method like this can of course never be better than the examples we trained on. For example, this paper here have taken sequences from a specific family and trained on that. And then they are of course reasonably good to predict the structure of sequences that looks like that. So but there is nothing in here that says that you cannot predict pseudoknots or any kind. So but again, it's probably not a general method. And also it's modeled on the fact that this is what people do when they do image recognition, right? And here in this input image, the larger it is, the more examples you need, because then essentially you have more input, right? And you cannot really compress it. A lot of image recognition thing works because not every pixel matters in an image. There's a lot of things that can be compressed. Here, every pixel kind of matters. So it's probably, maybe it turns out this is just a cute approach to it, because a lot of the other thing also works. You can handle large images, but the way you handle them is that you look at them piecewise.
So just one thing on that's because that we have discussed that this might be a way to make a big library of sequences that are not natural, but that's just RNA sequences, and then do a shape on those and get the secondary structure.
I think that would be extreme.
To the 9s of sequences or something like that. Like technically that should be
possible. I think that would be,
and then you could do that co-transcriptionally or even hit a needle and then do it twice. So that was just why I asked.
I think that many examples of this kind, right, would, not just took this picture, but would really be nice in order to both use machine learning based methods, but also for things that are more, whether you call them a machine learning or more traditional, you basically just have these examples. You also need such examples to validate your method. I think we have a few. But I think in this particular paper, they had a couple of thousand examples from a family
So
where they then trained it on some and tried it on others. But typically, machine learning cannot really answer something if you spend many years trying to find 10 examples. That's not enough to generalize from.
Because the structures that are from RNA in nature, that is actually the RNA structures that act like proteins, that actually has a structure that you have been looking at. So probably there's a lot of RNA structures that are really dynamic in the nature that's not in the structure databases that we won't see.
But I am working on answering your question more like in terms of numbers, but it's kind of hard. But I'm working on exactly that. Yeah.
And then there are many options. Johan also showed you this picture, but this is just another approach that does the same thing abstractly, but has a few. This step is done differently. But essentially the same. But there's tons of these things also big forward because they seems like low-hanging fruits to apply some existing methodology from image recognition, and then you get some results that actually might be useful. Then we have also been playing around trying to formulate and trying to get some CS student interested in a more, say, abstract COFOLD prediction problem, where you could say that could maybe appeal to computer scientists to say, could they make a model, you could call it a toy model or a toy solution, where maybe you could learn something about what actually makes such algorithms hard. Namely, where you don't look at the prediction problem as you are given the full sequence and then you predict the full structure. You maybe try to make an algorithm that looks at it like a sequential process. You have a sequence, you are given it piece by piece, where piece could be a single base pair, or it could be a few base pairs, a constant number of base pairs. And then given a first part of the sequence and a structure of you have up to that point, then you make a folding algorithm or a step in the folding process, where if you're given that, then you take the new information you have about the sequence, the existing structure you have about the information about the previous part of the sequence, and then you predict the structure for the full part of the sequence. And then a structure prediction would kind of be just run this sequence length time. And then you could be interested in such things as saying that a sequence don't have a structure for the full sequence. It basically have a sequence of structures or trace, namely the structures that is attained for each initial part of the sequences. So at least abstractly, that reminds me of the COFOLD or co-transitional folding process. Eva has explained to me that you kind of build this structure up piece by piece, and then how would it be in here? That should maybe be a score function or a mathematical function that takes into account that, okay, now you know some more, but it also maybe make a super good folding of the entire part, but you had to destroy what you already had. Maybe that's not as likely as if you did something else that wasn't as destructive as ripping all you had already apart. Could you do that? People have already done that, where they basically said, okay, now look that. Let's just take one existing folding, M-fold, RNA-fold, whatever, and then see how does the predicted secondary structure look like for the first 10 base pairs? How does it look for the first 20, for the first 30, or whatever step-by-step length they take? And then they get a sequence of structures for prefixes of the sequence, and they can maybe study that as a trait. But maybe instead of just ripping everything apart and refolding it, you could make a refolding algorithm and study that trace. I know people have done it before, but I still think it would be fun. I haven't really convinced any PhD or PhD for say master students in computer science or our own to do this. But you could make nice small animations, or maybe you could then learn something of how much it would be reasonable to assume that would happen during the wave, right? Yeah. And then I think as part of this project, we always wants to assist if you guys have any problems. We mention something, so that is if we could as a assistance there. And then I think the last slide I have is that what we have discovered, and mainly you, and that's of course, we already knew that, but data is really important. Many of these methods really needs a lot of data either to construct or to validate. And that don't really seem to be that. And you also then, as you already explained, that a lot of the data that people have used seem to be maybe biased in some way. Maybe they don't really have a representative set of examples, or the sequences are too similar, or and that probably reflects that many of the groups that do algorithms, folding algorithms, they of course care about the folding algorithms, the good ideas that goes into that, and the validation part is often maybe not what of course should have the interest, but they don't have the competencies to do it. And also where such methods are talked about and published, they care about the methodology and so. But you've been looking into the data, and you have added a few slides to that.
Yeah.
Just fast forward to the ones you want to talk about.
Yeah, but I think we kind of talked about the things. Like many of you have seen these slides already. That's why I'm like.
So Johan did a presentation at our group meeting some months ago.
But I think maybe this one is kind of relevant to you guys, because the thing is, many like these thermodynamic methods have been looking into these kind of sequential folding problems. These are two examples, and COFOLD is basically a heuristic method of just having helices opening and closing at pseudo randomly based on some Monte Carlo model. Interesting to look at, not really close to real life. And then there's Kinwalker, which is RNA folds model using, you set some limit to the thermodynamic barrier between what exists already and whatever structure you get or a new sequence you get. And if and only if that barrier is overcome or can be overcome, set by you as the user, it will fold onto that position. But it ends up in the minimum free energy structure, so it doesn't really like take that folding into account. It's just trying to simulate the folding from one end to the other, which is kind of like, it's almost what we want, but it's not quite. We kind of want to predict the ending using this step-by-step method. And I guess you can use like the energy landscape to predict this if you have some kinetic parameters, and we don't really have a lot of those. We don't know how long things take. We just know what is the best minimum free energy at each step, and we can calculate that pretty well with the RNA fold method, but it doesn't really tell us the whole story, and that's kind of the problem here. We need some kinetic estimate of a lot of things in RNA, how fast it happens, if we had to do a model that really kinetically walks through this, because I would argue that Kinwalker is not actually kinetic at all. It's just setting some limit on the thermodynamics, which of course represents some kind of kinetic, some kind of kinetics, but it's not really the barrier, the true barrier of things.
It doesn't get caught in a like
in some because it will always just look at what will it take to break these base pairs and these interactions between the base pairs and so on, all the thermodynamic parameters that is included in the model, and it will see if the new basis is enough to overcome that.